When you think of bees, you may think of little bees making honey or the painful sting they can give you. But when scientists think of bees, they think of autism. Allow me to explain. We will not be talking about honeybees today, however. We will be talking about sweat bees. Sweat bees are black and metallic colored and are attracted to the salt in human sweat. French biologist Cécile Plateau-Quenu noticed that they have different types of habitats, one being much like the honeybee in a hive, while the other were individual survivors. It was found that these traits were inherited, so one type of sweat bee was known as social, while the other was known as solitary. Later on, Sarah Coker at Princeton wanted to expand on this discovery by looking into the genomes of the two groups to find where their genetic differences were. Genome is a complete set of genes or genetic material present in a cell or organism. In order to compare genomes, scientists must collect the same short strand and decode the nucleotides to find what the RNA strand would be in order to identify the protein it would make. It was found that there were 200 differences within only 62 genes between the two groups, the most important one being syntaxin 1A. This gene codes for the protein syntax, which is an important aspect in the neurotransmissions between neurons or nerve cells, by promoting the movement of calcium ions. When they measured the amount of syntax produced by these two groups, they found that social bees had 15 times more syntax production than solitary bees. This increase makes the bees more likely to socialize, with their neurons shooting more impulses. This correlation can also be seen in insects that are only social during mating season. This increase is due to the other 97% of genetics that regulate which proteins are made rather than coding for proteins themselves. So when it is crucial for organisms to be social, the regulating genes will act like a light switch and call for more syntax production. Through comparative genome sequencing projects, it can be seen that syntax and regulatory genes that affect the social behavior of sweat bees also affect humans and can cause autism if there is too little of it present in the individual's genome. This further advances our idea that the social behaviors of organisms are evolutionary and partly built into their genetic makeup. Autism is a spectrum, so it has many different causes, but most of the time causes are unknown. However, it can be seen that it can either affect early brain development or the ways in which neurons interact with each other, much like the bees. So the next step is to investigate syntax levels in humans with autism versus those without. If a significant difference is found, we could begin to look into methods that can promote the production of syntax to help stimulate neurons in autistic people by turning on protein coding genes or base replacements in DNA coding. This increase in syntax could help autistic individuals be more comfortable in social situations and improve their ability to communicate with others. So now, what do you think of when you hear bees?